just picks up some pebbles, throws the pebbles into the cave to sort of scare the goats out. Except what he hears.
For 70 years, the greatest minds in biblical archaeology stared at 25,000 fragments of ancient parchment and tried to answer one question: who wrote the sea scrolls? They analyzed handwriting, they compared linguistic styles, they studied ink composition and scroll construction and every physical detail visible to the human eye. They built an entire theory, published thousands of papers, taught it in every university on earth, and shaped everything we believed about the origins of Judaism and Christianity around it. That theory held for 7 decades. Nobody seriously challenged it because nobody had the tools to prove it wrong. Then a molecular biologist sat down next to a Bible scholar on a bus and asked a question so simple it changed everything. What are the scrolls made of? The answer was animal skin. And the moment those two words left the scholar's mouth, the biologist realized something that would take another 7 years to prove, but that he already knew in that instant. The parchment had DNA, and that DNA was about to tell a story that 70 years of traditional scholarship had gotten fundamentally, catastrophically wrong. This is what the genetic fingerprint revealed. This is who it belonged to, and this is why the researchers who saw the results had to sit with them for months before they were ready to tell anyone what they'd found. In 1947, a Bedouin shepherd boy named Muhammad Ed-Dib threw a rock into a cave near the northwestern shore of the sea. He was looking for a lost goat. What he heard was the sound of something shattering. When he climbed inside, he found tall clay jars sealed with lids, sitting undisturbed in the darkness. Inside them, wrapped in linen and blackened with age, were leather scrolls. He had no idea what they were. He sold them to an antiquities dealer in Bethlehem for the equivalent of a few dollars. Those scrolls would turn out to be the most significant archaeological discovery of the 20th century, the Sea Scrolls. The oldest known biblical manuscripts in existence, dating back more than 2,000 years. Between 1947 and 1956, archaeologists and Bedouin treasure hunters found manuscripts in 11 different caves scattered along the cliffs near an ancient settlement called Qumran. The texts included the oldest surviving copies of books from the Hebrew Bible, alongside sectarian religious writings, community rules, hymns, prayers, apocalyptic prophecies, and commentaries on scripture that had never been seen before. They spanned roughly 3 centuries, from approximately 250 BCE to 68 CE, covering one of the most critical periods in the history of both Judaism and Christianity. The scrolls were remarkably well preserved, protected by the dry desert climate, and sealed inside those clay jars. But they weren't intact, far from it. What scholars had to work with was not a neatly organized library. It was 25,000 fragments, most of them tiny, some no bigger than a fingernail, many with only a few partial words visible on their surface. No titles, no page numbers, no labels telling anyone which pieces belong to which manuscripts. Imagine someone took 1,000 different jigsaw puzzles, ripped each one into hundreds of pieces, mixed them all together in a pile, threw away most of the pieces, and then asked you to figure out which fragments belong to which puzzle without ever showing you what the finished pictures looked like. That was the challenge facing Sea Scrolls scholars for the next 7 decades, and the stakes were not academic. If you placed 2 fragments together that didn't actually belong to the same manuscript, you could create false readings that would mislead every researcher who relied on your reconstruction. You could attribute ideas to ancient authors that they never wrote. You could distort the foundational texts of 2 of the world's largest religions. The margin for error was enormous. The consequences of error were severe. Using every tool available to traditional archaeology and textual scholarship, scholars built a theory that became the dominant explanation for who wrote the scrolls and why they were hidden in those caves. The Sea Scrolls, according to the consensus view, belonged to the Essenes, a Jewish sect described by ancient historians like Josephus and Pliny the Elder. The Essenes supposedly lived in a monastic community at Qumran, isolated from mainstream Jewish society, following strict purity laws and dedicating themselves to copying sacred texts. When the Roman army advanced through Judea during the Jewish revolt of 66 to 70 CE, the Essenes hid their library in the nearby caves to protect it from destruction. There it sat, undiscovered, for nearly 2,000 years. The theory was elegant. The sectarian writings describing community rules and initiation rituals matched what ancient sources said about the Essenes. The calendrical texts showing a solar calendar different from the lunar calendar used by the Jerusalem temple establishment suggested a group that had deliberately broken from mainstream Judaism. The apocalyptic writings expressing hostility toward the temple priesthood fit the image of a radical separatist movement waiting for divine intervention to set things right. For decades, this became the accepted explanation, taught in universities, presented in documentaries, printed on museum placards. The sea scrolls were the library of the Essenes, case closed. But there were problems that nagged at scholars even as the Essene theory dominated every discussion. The scrolls showed enormous textual variation, different versions of the same biblical books that contradicted each other in significant ways, fragments written in different scripts by different scribes using different writing conventions, parchment quality that ranged from finely prepared skins to rough, barely processed materials, and radiocarbon dating that suggested some scrolls were older than the Qumran settlement itself. Most puzzling was the diversity. If these were the private library of one isolated sect, why would they possess multiple conflicting versions of sacred texts? Why would they preserve writings representing theological positions they fundamentally disagreed with? Why would a community copying text for its own use according to its own specific worldview maintain that kind of variety? These questions had no satisfying answers within the Essene framework, but without hard evidence to support an alternative, the established theory held. It was the best explanation available given the tools scholars had. Then, in 2012, everything changed because of a conversation on a bus. Oded Rechavi and Noam Mizrahi were both new faculty hires at Tel Aviv University. They sat next to each other during an orientation event, a bus ride between campus buildings. Small talk, introductions. What do you study? Mizrahi was a biblical scholar specializing in ancient Hebrew literature. He studied the sea scrolls and understood at a granular level every textual puzzle that had frustrated researchers for decades. The fragments that might belong together but couldn't be proven. The manuscripts that scholars had assembled based on educated guesses about handwriting similarity. The enormous risk of getting those guesses wrong. Rechavi was a molecular biologist. He studied genetic inheritance in nematode worms. He had no background in biblical studies, no training in ancient languages, no connection to archaeology whatsoever. But when Mizrahi explained that the scrolls were written on animal skin, Rechavi's mind went somewhere no biblical scholars had gone before. Animal skin contains DNA, even skin that has been soaked, scraped, stretched, dried, written on with corrosive iron-based ink, and left in a desert cave for 2,000 years. The DNA would be degraded, fragmented, contaminated beyond recognition by modern handling, but it would be there, embedded in the collagen fibers of the parchment itself, waiting. If they could extract it, sequence it, and compare it across fragments, they could do something no amount of handwriting analysis or textual criticism had ever been able to do. They could prove, at the molecular level, which fragments came from the same animal, which animals were related to each other, which parchments were produced in the same location, and which ones came from somewhere else entirely. It was the kind of idea that sounds obvious in hindsight, but that nobody in 70 years of Sea Scrolls research had ever seriously attempted. The project that followed would take 7 years. It would involve researchers from Tel Aviv University, Uppsala University in Sweden, Weill Cornell Medicine in New York, and the Israel Antiquities Authority. It would require the development of entirely new methods for extracting ancient DNA from sacred texts without damaging a single letter of the writing on them. And what it revealed would stun the archaeological world. The first challenge was extracting DNA from manuscripts that are considered sacred by multiple religious traditions and are so fragile that any destructive sampling was absolutely forbidden. You cannot cut a piece off a sea scroll. You cannot drill into it. You cannot remove material from any portion that contains text. The scrolls must be preserved intact for future generations. So how do you extract DNA from something you are not allowed to touch? The team found a solution that was equal parts brilliant and absurdly simple. Over decades of handling and study, microscopic particles of parchment had accumulated as dust in the storage containers where scroll fragments were kept. This dust contained collagen, the structural protein that makes up animal skin, and embedded within that collagen was ancient DNA. By carefully collecting dust from blank margins and damaged edges where no text existed, the team could gather enough genetic material for sequencing without touching a single word on the scrolls. They were extracting secrets from material that had been treated as waste. Particles so small they would have been swept away and discarded by anyone who didn't understand what they contained. Scroll dust. 2,000-year-old residue from the backs of fragments that nobody had thought to examine. But getting the DNA out was only the first problem. The parchment-making process itself, the soaking, scraping, stretching, and drying of animal skins, damages DNA at the molecular level. Then the skins were written on with iron-based inks that are chemically corrosive, further degrading the genetic material. Then they sat in caves for 2 millennia, exposed to temperature fluctuations, humidity changes, and mineral deposits. And then they were handled by hundreds of people over 70 years, each person potentially leaving traces of their own modern DNA on the fragments. The result was genetic material that was fragmentary, severely degraded, and buried beneath layers of contamination from every human who had ever touched the scrolls. The team worked in specialized ancient DNA laboratories with the strictest contamination controls in the world: full protective suits, masks, gloves, positive air pressure to prevent outside air from entering, ultraviolet sterilization between every procedure, every step documented and controlled. They extracted human DNA first, identified it, and removed it from the dataset. They looked for molecular damage patterns characteristic of ancient DNA, specific types of breakdown that occur only over centuries, to authenticate what remained. They cross-checked results between separate laboratories to ensure reproducibility. They developed statistical methods to distinguish ancient sequences from modern contaminants with absolute certainty. It took years, hundreds of samples, thousands of hours of laboratory analysis, careful validation of every single result, and then the sequences came back, and the room went quiet. The first finding seemed almost too simple to matter, but its implications were seismic. Most of the scroll fragments tested were made from sheepskin. That in itself was a surprise. Scholars had previously assumed the scrolls were written primarily on goatskin. They were wrong. But sheep were common in ancient Judea, and sheepskin was a reasonable material for parchment production, nothing earth-shattering yet. Then the team found something that was. Several fragments were made from cowhide. That single biological fact, cow versus sheep, detonated the foundation of 70 years of established theory. Here's why. Qumran sits in the Judean desert near the sea. It is one of the harshest, driest environments in the entire region, a landscape of rock, sand, and sparse vegetation. Water is scarce, grazing land is minimal. Sheep and goats can survive there because they're adapted to desert conditions. They require relatively little water and can graze on tough, sparse plants. Cattle cannot survive in that environment. Cows need abundant water, dozens of gallons per animal per day. They need substantial grazing land with actual grass. They are not desert animals. There was no way a community living at Qumran could have raised cattle. They could not have had regular access to cow hides for making parchment. Any scroll fragment written on cow parchment came from somewhere else, somewhere with water, somewhere with pasture, somewhere outside the desert. The DNA proved, beyond any possibility of doubt, that not all the sea scrolls were produced at Qumran. The core assumption of the Essene theory, that these were the private library of one isolated desert sect, could not survive this finding. But the cowhide was just the beginning. One of the most significant discoveries involved 2 fragments that scholars had debated for decades. Both appeared to be from the book of Jeremiah, but the versions of Jeremiah they contained were dramatically different. One version was close to the Hebrew Bible as it exists today, longer, with chapters arranged in a familiar order. The other version was significantly shorter, missing entire sections, with chapters arranged in a completely different sequence. Scholars had argued for years about whether these represented the same scroll that had simply been damaged differently, or whether they were genuinely separate manuscripts reflecting different textual traditions. The DNA settled it permanently. One fragment was written on cow parchment, the other was written on sheep parchment. They came from different animals, almost certainly from different regions, and they could not possibly have been part of the same scroll. Two different versions of the book of Jeremiah, written on different animal skins, brought from different geographic locations, preserved side by side in the same cave. The differences between them were not copying errors or scribal mistakes. They were genuinely distinct versions of sacred scripture that had been circulating simultaneously in different communities across ancient Judea. That finding alone forced scholars to reconsider everything they thought they knew about how biblical texts were transmitted and preserved in the centuries before the destruction of the Second Temple. There was no single authoritative version. There were multiple competing versions, maintained by different communities, each considered legitimate by the people who used them. The team went further. By analyzing nuclear DNA markers, they could sometimes determine whether parchments came from animals that were genetically related, siblings from the same flock, parents and offspring, cousins, animals that were raised together in the same herd at the same location at roughly the same time. When they found clusters of genetically related parchments, it indicated a production center where multiple scrolls were being manufactured from locally sourced materials. And when they found parchments with no genetic relationship mixed in alongside those clusters, it indicated that scrolls had been brought in from elsewhere. The pattern was unmistakable. The C scrolls were not the product of one community working in isolation. They were assembled from multiple production centers across ancient Judea. Some fragments came from local sheep near Qumran. Others came from completely unrelated animals living in distant flocks. The cow parchments came from regions with enough water and pasture to support cattle far from the desert. The genetic evidence supported a theory that some scholars had proposed but never been able to prove. The caves near Qumran may not have been the hiding place of one sect's private library. They may have functioned as a Geniza, a sacred storage place where Jewish communities deposited worn-out or theologically problematic texts that contained the divine name and therefore could not be destroyed according to Jewish law. Jerusalem is only 20 miles from Qumran. When the Roman army besieged the city between 68 and 70 CE, refugees fleeing the destruction could have brought sacred texts from multiple synagogues and communities to the caves for safekeeping, adding them to materials that had already been stored there over previous decades. This would explain the enormous diversity of the collection. It wasn't one group's theological statement, it was the accumulated religious literature of multiple communities across an entire nation, preserved together when the final crisis came. Consider what the DNA told them that 70 years of traditional scholarship could not. The scrolls came from multiple locations. Multiple communities had been copying scripture independently, maintaining different versions they each considered authoritative. The biblical text was not monolithic or uniform during the Second Temple period. There was no single universally acknowledged correct version that everyone followed. The process of deciding which version of a sacred text was the definitive one was still ongoing, still contested, and still unresolved when these scrolls were hidden in those caves. That has profound implications for understanding the development of both Judaism and Christianity. The diversity preserved in the C scrolls reveals the theological ferment of the exact period when Christianity emerged from Judaism. The debates over scripture, law, messianic expectation, purity regulations, and community organization reflected across these fragments provide crucial context for the world in which Jesus lived and in which the early Christian movement took shape. And none of it would have been discoverable without reading the genetic code embedded in the skin of animals that died 2,000 years ago. Professor Rachavi put the significance of this in terms that no biblical scholar could have formulated. Imagine that Israel is destroyed to the ground and only one library survives, the library of an isolated extremist sect. What could we deduce, if anything, from this library about Greater Israel? That was the question the DNA answered. The C scrolls were not the library of one sect. They were a window into the diversity and richness of Jewish religious life across an entire civilization at one of the most important moments in human history, and it took reading the genetic fingerprints of sheep and cattle that had been for 2 millennia to finally see it. The team has only sampled a fraction of the 25,000 fragments. Thousands remain unexamined. Researchers are already expanding the study, building databases of ancient animal DNA from different regions of the ancient Near East, creating genetic reference maps that might eventually allow them to pinpoint exactly where specific parchments were produced. They're combining genetic data with multispectral imaging technology that can read text invisible to the naked eye, ink analysis that can identify chemical signatures of different scribal workshops, and radiocarbon dating refined to narrower time windows than ever before. Every fragment they test adds another data point to a map that is slowly revealing the full picture of where these texts came from, who produced them, and how they moved across the ancient world. The scrolls have been sitting in climate-controlled vaults for 70 years. Scholars thought they had extracted everything those fragments had to give. They were wrong. The parchment itself was holding information that no human eye could see, a genetic record written in the collagen of animal skin that preserved the biological identity of creatures that grazed in fields that no longer exist, in communities that were destroyed 2,000 years ago. That record waited, patient, silent, encoded at the molecular level in material that everyone assumed had nothing left to say. And when scientists finally learned to read it, the story it told didn't just add a footnote to what we knew. It rewrote the entire chapter. What do you think the remaining fragments will reveal when they're finally tested? Tell me in the comments, and if you want to know the moment the next round of DNA results surfaces, hit subscribe and turn on notifications, because what's hidden in those scrolls is far from fully decoded.